Dear students, now let us move to the next question. Question number 6. Here, there is a table which gives about the idea about different stains. Different stains and their uses and final color is given below. Choose the correct option. Because you should have the basic awareness about the stains, because definitely stain related question I have seen in some question papers. That's why I said this particular question. In the stain column, option A, basic fuchsin used in staining of bacteria, and the color is violet. The slide will show violet color. Then fuelgen stain, it is used to color the cytoplasm, and the slide, the material will take pink color. Then acid fuchsin, it is a fungal hyphae, and the final color is blue. Safranin is a stain used for staining is nucleus, and final color is red. Question is, different stains, their uses and final color is given below. Choose the correct option. Here, whatever the options they are given is not 100% correct. Only one option is correct. So you have to find out which one is correct. For that, you should have the basic knowledge of stains. So, for that, the option, let us move. Option is D. D is proper one. Option, question number 6. Option D. Option D. And I will write a chart of different stains and uses and a color. You write down in your short notebook. So all the whenever you make a short note, you should make it in such a way that you should be able to carry it to the exam center, where you can just revise it during the time before half an hour of the exam, or while going you can have that revision. Okay. Acetocarmine is one stain used to stain the chromosomes. What is the color? Chromosomes. We can analyze by observing the slide which is the stain by seeing the color, pink color. It takes which color? Pink color. Second one, acid fuchsin. Acid it is used to stain cortex and mitochondria. Cortex and mitochondria. Cortex of the plant, and color is magenta. Magenta color. Then aniline blue. It is fungal hyphae, used to stain fungal hyphae. And the color is blue as a name. It is very easy to remember the color. Okay. Then fourth one, basic fuchsin. Basic fuchsin is another stain. It is used to stain the nucleus. It is used to stain the nucleus. And the color is magenta red. Magenta red. This color will be a little bit darker than the previous one. Then crystal violet is the name of the stain. Crystal violet. And it is used to stain the bacteria. And the color is violet. Easy to remember. See. And sixth one, eosin stain. It is used to stain the cytoplasm, and the color is pink. There will be a little bit slight variation. Eosin is always dark pink. This is comparatively light pink. Then fuelgen stain used to stain the DNA, and its color is purple or red. So this is the information that I can give you with regard to stains. Few more is here. Eighth one. Hematoxylin. Dear students, it is used to stain nuclei and cell wall. It is used to stain nuclei and cell wall, and it gives violet color. Then ninth one, iodine solution used to detect the presence of starch in the given sample. Blue color. Then 10th one, so Sudan black is the name of the stain. Sudan black. And it is used to detect the fatty substances which are present in the given tissue substance, and its color is black. If a tissue which is taken, if it gets black patches, then it's an indication that the fatty substance is there. Then toluidine blue. And it is used to stain RNA, and the color is blue. I think I have given almost all the stains, and the place where we use to stain the cell parts I have given in the middle column, and indication color is also given. So next question, number 7. Find out the wrong statement with respect to cell organelles from the following. Lysosomes are the single membraned vesicles budded off from Golgi apparatus and contain digestive enzymes. Then endoplasmic reticulum consists of network of membranes, tubules, helps to transport and synthesis and secretions. Leucoplasts are bound by two membranes, lack pigments, but contain their own DNA, protein synthesizing machinery. Spirosomes are single membrane bound and are associated with synthesis and storage of lipids. So I have given 4 options here. So the answer for this is 7th question. Answer is C. Leucoplast means you studied it, colorless plastids. Then there are 3 types in leucoplast. What are the 3 colors? Amyloplast, then proteinoplast, and then elaioplast. Okay? And amyloplast, synthesis and storage of starch. Okay? Then it helps in storage of protein. And elaioplast stores lipids. So These are all some of the important informations that you should remember. Question number 8. Read the following statement A and B. Statement A, the number of mitochondria in a cell do not correspond to the functions of the cell. Statement B, mitochondria are common to both plant and animal cells. So choose the correct option. Statement A is correct, B is wrong. Statement B is correct, A is wrong. Both the statements are correct, both the statements are wrong. So option 8, the correct answer is option B. Question number 8. Answer B. Answer B. Because mitochondria takes part in, usually it is option B what it says. Statement B is correct, A is wrong. Because the number of mitochondria in a cell always correspond to the function. For example, in our leg, the muscle cells will have around 3000 mitochondria because of strenuous work, because they have to provide energy for the muscles during running, walking, everything. And that's why always the number of mitochondria depends upon the work which is done by the cell. So mitochondria, it is involved in photophosphorylation and Krebs cycle, aerobic respiration, and number depends upon the cellular activity, and the number of mitochondria and site, the place. It is 1 in chlorella. Chlorella, one cell possesses only 1 mitochondria. And 25 in sperm. Sperm Nebenкern. It is about 50,000 in giant amoeba. It's about 5 lakh in flight muscles of birds. Flight muscles. It is 1 in chlorella, 25 in sperm, 50,000 in giant amoeba, bigger amoeba, 5 lakh in flight muscles. So, depending upon the activity, the number of mitochondria, it varies. That you should remember. Okay? Now, I'll continue. The next question number 9. What it says, question number 9. See, they have given a diagram and they mentioned different parts of the nucleolus, and you have to find out the correct answer. For that, the correct answer is option D. Option D. 10th question. Nothing is there to explain here. Euchromatin and heterochromatin, that you should remember here. Then in the diagram, some parts are labeled with alphabets, and match them and choose the correct answer. So here also answer B. B is the answer for this question. And chloroplast, it is a 2-layered, and outer membrane and inner membrane. And here you should remember that this chloroplast has 2 layers, outer layer and inner layer. The inner content is called stroma. This is the outer layer, and here is inner layer. And my dear students, here you can see the presence of coin-like structures which are arranged one above the other. They are called Thylakoids. What are these? Thylakoids. Thylakoids. And here the small structures will be there to trap the sunlight. They are called quantosomes. Quantosomes. And this whole structure is called grana. And they are interconnected with another grana by stroma lamella. Lamella means layer. Stroma lamella. This is stroma lamella. Lamella means layer. Stroma lamella. So now I will move to the next question. Choose the correct matching. Cytoplasm inside the cell, protoplasm except nucleus. Cytoplasm outside the cell is called mesoplasm, hyaloplasm, kinoplasm, cytosol. So cytoplasm inside the cell is called protoplasm except nucleus, because protoplasm means question number 11. Protoplasm anta helidre, protoplasm means nucleoplasm and cytoplasm. Cytoplasm plus nucleoplasm, not nucleus. Solid content will not be there. Then cytoplasm outside the nucleus is called hyaloplasm. Option B, hyaloplasm. Outside the cytoplasm, outside the cell. Next, question number 12. Question number 12. I'll move to question number 12. Question number 12. 80 S ribosomes has, S means Svedberg unit, Svedberg constant. 3 molecules of ribosomal RNA, 16S, 5S, 23S and 50 protein molecules. 4 molecules of ribosomal RNA, 5S, 5.8S, 16S, 28S and 80 moles of protein. 4 molecules of ribosomal RNA, that is 5S, 5.8S, 16S, 28S and 70 moles of proteins. 4 molecules of ribosomal RNA, 5S, 5.8S, 16S, 28S and 60 molecules of protein. For this question, answer is B, option B. Answer B. That is 70S ribosome. Then ergosomes. Here I want to introduce a new term. Ergosomes are also called polyribosomes. Polyribosomes means long chain of ribosomes. Polyribosomes. They are called ergosomes. Okay. So 12th question, option is B. Now let us move to question number 13. Question number 13. Question number 13. Here the question is, the increasing order of plastid size. We have to identify which is smaller and which is a bigger one. Increasing order. That means we should start from smaller to bigger one. Option A, chromoplast, chloroplast, elaioplast, aleuroplast, amyloplast. Option B, amyloplast, aleuroplast, elaioplast, chromoplast, chloroplast. Option C, chloroplast, chromoplast, elaioplast, aleuroplast, amyloplast. Option D, elaioplast, aleuroplast, amyloplast, chromoplast, chloroplast. So we have to find out that the correct order is option C. Option C. Option C. Then 14th, M chromosomes are found in. M chromosome means I think you didn't use this particular word. M means minute sized chromosome. Very small chromosomes. Minute sized chromosomes. M chromosomes. Minute sized chromosomes. Minute sized chromosomes. They are called M chromosomes, minute sized chromosomes. Okay? Then what they are asking, M chromosomes, bryophytes and insects, pteridophytes and insects, angiosperms and molluscans, gymnosperms and molluscans. So the answer is M chromosomes are found in option A, that is bryophytes and insects. A chromosome, chemical component of chromosome. This is important for your, they may ask a question. Chemical component of chromosome. Chromosome. 40% DNA. Then 50% histone protein. And then 15% RNA. 15% RNA. There in molecular biology, we used a term that is called trabant. Trabant. What is trabant? It is a new word for you. Trabant means satellite, or satellite means chromosome consists an extra point that is called satellite, and that is also called trabant. Trabant, that word you should remember. And in heterochromatin there are two types. Heterochromatin, there are following different types. I will give you. Facultative. That means temporarily inactive. Temporarily inactive. And constitutive. Constitutive means it is permanently inactive, or it is also called heterochromatin. Permanently inactive. So usually it is found near centromere. Found near centromere. Here they can ask a question, which of the following component of chromosome is correct? Option A, 50% DNA, 40% histone, 15% RNA. Option B, 15% DNA, 40% histone, 50% RNA. Like that, by shuffling it, they may ask another question. So, better to remember this particular concept. So, I will move to another question. Question number 15. The correct order of sedimentation of subcellular structure during different centrifugation. Sedimentation means, depending upon the weight, they go and deposit in the test tubes. If a component deposit at the bottom, it's an indication that it's a heaviest particle. Suppose during centrifugation, they use the test tube like this. If a particle is having more weight, it will deposit at the bottom. If the particle is having lighter weight, it will stick to the wall of the test tube at the upper end. So depending upon that, we can assess the weight of the object. So the question is related to that. What they are asking, the correct order of sedimentation subcellular structure during differential centrifugation. Option A, lysosome, mitochondria, nucleus, ribosome. Option B, mitochondria, nucleus, ribosome, lysosome, ribosome. Option C, nucleus, mitochondria, lysosome, ribosome. Option D, lysosome, ribosome, mitochondria, nucleus. Here the answer is C. Answer C. What's the meaning of this? The correct order of sedimentation of subcellular structure during differential centrifugation. First nucleus sediments, that is having more weight, and after that mitochondria, then lysosome, at the top ribosome. Then I think I'll continue this in the next part.